Okay, so today we're going to talk about a technique called capillary electrophoresis. Now, I know it's not exactly a household name, right? But what if I told you it was the secret key that unlocked one of the biggest scientific challenges of our time? We're talking about mapping the entire human genome. Yeah, it's that important.
So, to really get why this was such a huge deal, we've got to take a step back and look at the absolutely massive problem scientists were staring down.
I mean, think about this for a second. How on earth do you sort 3 billion DNA base pairs? And not just sort them, but do it with near perfect accuracy. This wasn't some thought experiment. This was the core challenge of the Human Genome Project. Scientists had to find a way to separate and pinpoint millions of tiny DNA fragments, fragments that were sometimes different by just a single letter of code. The tools they had, well, they just weren't going to cut it, not even close. So the old way of doing things was this thing called slab electrophoresis. And honestly, it was slow, it was a ton of manual labor. You could think of it like trying to sort individual grains of sand on a beach by hand. It was just brutal. And then along comes capillary electrophoresis, or CE for short. And this thing was a complete rocket ship. It was fast, it was automated, and it was unbelievably precise. It was, no exaggeration, a total game changer.
All right, so how does this miracle technology actually work? Well, the best way to think about it is to imagine a high-speed race, but instead of cars or runners, the competitors are molecules. And the racetrack? It's literally thinner than a single human hair. Yeah, that's really the heart of it. CE is basically a supercharged microscopic racetrack for molecules. It separates them by pitting them against each other inside this tiny, superpowered arena. And whoever wins, or where they place in the race, that tells us exactly what's in our sample.
And when I say microscopic, I am not kidding. This racetrack we're talking about is a tiny little glass tube, a capillary, with an inner diameter of around 50 micrometers. To put that in perspective, that's about the width of one of your hairs. And this incredibly tiny scale, it's absolutely crucial for making the whole thing work so well. Okay, so what about the supercharged part? Well, get this. We're blasting up to 30,000 volts of electricity across that tiny, hair-thin tube. That massive electric field is what acts like a rocket engine, pushing the molecules at unbelievable speeds. And that's what makes the separation both lightning fast and ridiculously precise. All right, so we've got our racetrack and our power source. Now let's get into the rules of the race. What actually determines who wins? So the basic idea here is something called electrophoresis. It's actually pretty simple. If you take a charged molecule, an ion, and you stick it in an electric field, it's going to move. Positive ions zoom towards the negative end, and negative ions race towards the positive end. Makes sense, right? Opposites attract. So what about neutral molecules with no charge? You'd think they'd just sit at the starting line, totally unaffected. But as we'll see, there's a really cool twist to that story.
So a molecule's speed isn't just random, it's all about its charge to size ratio. Think of it like this. The charge is the engine, and the size is the weight. A molecule with a big engine and a light frame, so lots of charge and a small size, is going to be the fastest. It'll just zip right through the liquid inside the capillary. And that right there is the core of how we tell everything apart. And let me tell you, this system is ridiculously effective. In science, we measure how good a separation is in something called theoretical plates. It's a weird term, but just think of it as a score. The more plates, the sharper and cleaner the results. Now, another really common technique, HPLC, might get you about 10,000 plates. That's pretty good. But capillary electrophoresis, it can blow that away, generating 150,000 plates, sometimes even more. What that means for a scientist is you get these beautiful, sharp peaks in your data. It's like going from a blurry old photo to a crystal clear 4K image. Just fantastic resolution. But wait, there's more. There's another layer to this whole thing. Kind of a secret weapon that makes CE so uniquely powerful. It's this wild phenomenon called electroosmotic flow. And the secret here isn't in what we put in the tube, it's the tube itself, the racetrack. See, the inner wall of that glass capillary, it naturally has a negative charge when it's filled with the buffer solution. And this one simple fact creates an absolutely amazing effect that changes the entire race.
So here's how it goes down. First, that negatively charged glass wall starts attracting all the positive ions from the liquid inside. They just line up along the wall. Then, when we flip the switch and apply that massive electric field, it yanks that whole layer of positive ions toward the negative end, the cathode. And since all those ions are surrounded by water molecules, they don't go alone, they literally drag the entire bulk of the liquid with them.
What you get is this powerful, steady current flowing through the capillary. The perfect analogy is one of those moving walkways at the airport. And because this flow is so incredibly uniform, it's not like a turbulent river, it's a smooth, flat flow. It doesn't mess up our separation. The little bands of molecules stay nice and tight, which is a huge reason why we can maintain that incredible, super sharp resolution. And here's where it gets really, really cool. This moving walkway is usually so fast that it's stronger than the individual swimming speed of any of the molecules. So think about this. Even the negative ions, the ones that are trying to run in the opposite direction toward the positive end, they can't fight the current. They get swept along with everything else towards the finish line. It's just amazing. And because of this, we can see and separate everything, positive ions, neutral molecules, and negative ions, all in a single experiment, and they come out in a perfectly predictable order. It's just so elegant.
Okay, so we've got this incredibly elegant, super powerful tool for sorting molecules. But the big question is, so what? What has it actually done for us out here in the real world? And this brings us right back to where we started, the Human Genome Project. The sheer speed and automation of capillary electrophoresis were absolutely essential for sequencing our DNA. I mean, it turned a project that honestly could have taken decades into something that was finished in just a handful of years. This was the engine that powered one of the greatest scientific achievements in modern history. But its impact goes way beyond just the genome. Think about the pharmaceutical industry. CE is a critical tool for making sure new medicines are pure. It can separate the main active ingredient from any tiny little impurities or side products, which is obviously crucial for safety. It even affects our daily lives. CE is used all the time to analyze our food. It's checking for things like additives, vitamins, and also the bad stuff, like contaminants or pesticides. It plays a huge role in keeping our food supply safe. And then there's forensics. You can imagine how valuable a tool that can analyze tiny, tiny samples with incredible precision would be. It's used to analyze all kinds of evidence from crime scenes. It even played a major role in the heartbreaking task of identifying victims after the World Trade Center disaster. It's just an invaluable tool. So when you boil it all down, capillary electrophoresis is just a beautiful example of taking a simple rule of physics, charged things move in an electric field, and combining it with some really clever microscale engineering. And the result? It completely revolutionized science and industry. It really leaves you with a fascinating question, doesn't it? We managed to decode the entire human genome using a racetrack the width of a hair. So what other huge, seemingly impossible challenges are out there, just waiting for the right tiny microscopic solution? What's the next big thing we're going to discover?